Hi, my name's Coen. I'm presenting my master's thesis presentation on the impact of surface asperities on optical coherence elastography. My supervisors were Dr. Brendan Kennedy and Dr. Chi Fang from Bright Lab. So I'll just get straight into it. And now the clicker's not working. All right. Lovely. So breast cancer has the second highest incidence rate in Australia, and most women tend to prefer having breast conserving surgery over mastectomies for treatment due to the lack of body disfiguration from the breast conserving surgery. The problem with that is that cancers tend to recur more likely, about 4 times more likely during breast conserving surgery over radical mastectomies. So why is that? One of the proposed reasons is that it's inadequate removal of the tumour. So the way that the surgeon will assess the complete removal of the tumour is by manual palpation. So they just press around within the cavity to see if to check that the tumour has been completely removed. There's a couple of problems with that. Firstly, that there's low spatial resolution of palpation and it's not objective, so it actually changes from surgeon to surgeon. Is there a way that you can utilise the properties of tumours to come up with some imaging system to try and better assess these margins during surgery? So the most important, the property that, the mechanical property that's assessed in palpation is stiffness. And so you can see here that stiffness is characterised by Young's modulus, which is the ratio of force over the deformation of that material. And you can see here that breast tumour has a significantly higher range of stiffnesses over the healthy tissue within the breast. One of the proposed methods of tumour assessment was optical coherence tomography, which is based on interferometry, so it works based on, the interference of light. So a laser beam goes into a beam splitter, it goes to the sample and the reference arm, and then it's reflected back, and the interference pattern is picked up by a detector, and the signal is at a specific distance. So if we change the length of the reference mirror, or use a broad beam light, so across a range of wavelengths, we can actually probe within the tissue and come up with a 3D image. And this was done in 2009. So Nguyen et al. Did intraoperative OCT assessment for tumour margins. And they found that you could actually tell tumour in the margin here, and it was confirmed by histology. The method I use is built on OCT. It's optical coherence elastography, which provides an easier to interpret image than an OCT system. So one of the limitations with OCT is that you might have to have specifically trained readers, like in sonography or histology. So we'll talk about how optical coherence elastography works. So when we talk about how, there's one in between. Sorry. How to describe elasticity within a material. We use the generalized Hooke's law, which relates the strain to stress by a 4-dimensional tensor, C. C has 81 independent parameters to describe elasticity, and we can reduce that to 2 by making assumptions based on mechanical homogeneity and isotropy. And then the final assumption for optical coherence elastography is that when you compress a material, it just moves in the one direction. So that Poisson's ratio is 0. And so it reduces the number of parameters required to describe the elasticity to 1. With strain imaging, so when you take an optical coherence tomography image, you get a phase array, a phase image based on the Fourier transforms. So this is randomly distributed through the sample from 0 to 2 pi, but is temporally invariant. So it means if you take scans of the same place over and over again over time, nothing changes. But if you apply a small perturbation, so you move the material slightly, the phase changes a little bit. The change in phase can be linked to the displacement of the materials within the sample, like by this equation, and so we can come up with a measure of strain or local deformation within the material. So the way that this is done for my project is that we have a rigid plate with a sample on it. This is the beam that goes to the OCT system. There's a transparent window that's placed onto the sample with a small actuation onto the sample, and so we take scans before and after compression to measure the displacement within the sample. This was done on excised breast tissue in 2016. So here you can clearly tell the difference between tumour and adipose tissue in the strain image. Now, strain imaging is inherently qualitative. It changes depending on how much force you're applying. So one of the ways to improve on that was suggested is quantitative elastography. So if we apply a transparent sensor layer before the sample with known mechanical properties, we can come up with a measure of how much force or stress is applied across the surface of the tissue. And if we then assume that that stress is constant at depth, we can work out the elasticity of the Young's modulus or elastic modulus within the sample itself. So this is the calibration curve for the sample. So we apply a preload stress and then a small perturbation, and then you can work out the perturbation stress at a specific point, and then the measured strain then is related back. And this was done in 2018, and you can clearly see that it's a lot easier to tell the difference between tumour and adipose tissue, even more so than in the strain image itself, by adding just a bit more definition with the colours. So one of the assumptions that we make in these images is that the stress applied is perfectly uniaxial, and so all the deformation is also just uniaxial. Now this clearly breaks down in situations where there's roughness in the surface, which is in surgical cavities. So then what happens is if you press down on a point, there's compression within the air over here, and so there's lateral expansion of the tissue. So that's what my project is sort of investigating, how these sort of effects impact the quality of these images. So overarching question, how do these surface asperities affect the quality of the images generated in both strain imaging and quantitative elastography? So take a systematic approach and look at both amplitude and spatial frequency in these across a range, and then we'll see how that affects the quality. These are the specific research questions. So both in strain and quantitative imaging, what is the effect of amplitude or peak size across these images? What effect does spatial frequency have? And with strain imaging, I also look at the difference between using a compliant layer and not a compliant layer. The compliant layer may more evenly distribute the force throughout the sample, so it might change the quality of our image. All right. So I made 3D printed molds. These were designed in SolidWorks. The accuracy of the molds is within 5% of the planned designs. They're mechanically homogeneous phantoms with chosen to have similar elastic properties to soft normal tissue. And it was the same material was used for the phantom and the compliant layer to minimize the impact of friction between them. Titanium oxide scatterers are suspended in the phantom to increase the OCT signal. So how it worked is I actually injection molded them. So this part sat onto the rest of the mold here, and the microscope slide was inserted across, and then the mold was held at an angle as the material was injected to allow escape of air. So it was a completely uniform phantom with a flat base. I made 21 phantoms with amplitudes ranging from 0 to 750 micron, and wavelengths from 0 to 4 mil. For each of these phantoms, I did 6 scans total at different preloads of from 10 to 60%, both with and without a layer. This resulted in 252 scans, and then subsequently there's at least 4 images generated for each of these scans: the OCT image, the phase difference, the strain image, and the quantitative image. So the 500 micron wavelength phantoms were found to be not structurally sound. So as you see, these get compressed, there's sort of air pockets that fall within them, and so that I thought, oh, that seems to introduce these extra artifacts, so they were excluded from analysis. This is the imaging system. So there was on the base plate here, it was lubricated first, the phantom was placed on it, and then you can see this compliant layer put on top of it. There was lubrication between every interface. The translation stage gets raised up to be, so the phantom or the compliant layer is just in contact with the imaging window, and then the preloads of 10 to 60% are then applied. 50 B scans were taken of each phantom, and then average. And so all of the strain and quantitative images are measured at the same time. Processing the images afterwards, I had to use edge detection to find the interface between the layer and the phantom and the oil in between. Oh, no. Oh, okay. So these red parts shows the region of interest, and these were determined based on specific, like you could see points where the force was directly being applied, because any sort of air gap introduces compressible air, and so then we can't assume that the force is going to be equal. And each depth is 500 micron from the interface between the layer and the phantom. The processing metrics used to assess quality. So strain imaging is inherently qualitative, and so the signal to noise ratio was used as the main metric, because in a homogeneous phantom, we are assuming that the mean should be equal because it's mechanically the same throughout the material. And we assume no knowledge of the forces across the region. For quantitative imaging, we're assuming that the known stress, the known elasticity here, should be exactly the same as the known elasticity here, because the forces are the same. So the strain shouldn't change, and so the elasticity should be the same. So what I did was look at the relative difference between each point vertically compared with one of the voxels above it. And so over the whole region, the mean difference between the measured elasticities from each each interface point and below it should be 0. So let's just get to the results. So this is just a uniform, flat phantom that I looked at. There's this imaging artifact that pops up, and this is just due to the OCT system. And so when analysis was done across this region of interest, again 500 micron, it actually gets removed because it does fall within the region of interest for some of the images. Let's take a look at one of the large asparity phantoms. So first consider the pink line across the top. Oh. No. Across the top, you actually see that at the sides, there's a large magnitude of strain. Again, strain is negative in this case, so lower down is a larger magnitude. And then at the center, it sort of gets up close to near 0. This is likely due to just a lot of freedom around the edges of the phantom, so it tends to move quite a lot. And you see that actually disappears after some time. If we take the blue and green scans, if we model the sample as a mass of springs and masses, you'd see this sort of, expect to see a concave up shape as we sort of move along in measured strain. That's exactly what you see and it doesn't seem to vary too much as we get down in depth. So it looks like the assumption of equal displacement seems to hold at depth. Finally, we look at the axial depth scan in the center of the peak, and you see that strain actually decreases over time in the plateaus. It's quite noisy, but This is due to friction, so there is some friction between the imaging window, in this case, and the phantom. And so it limits this lateral expansion, and because it can't move laterally, then it seems to struggle to move axially as well, because it does actually expand. This is just showing how at 10, 40, and 60% compression, the region of interest actually increases over time, over preload. All right, so this is going back to the fundamental research question. So what is the impact of amplitude on strain imaging? You can see that at low preloads, as we sort of increase the height of the amplitude, there seems to be a decrease in signal-noise ratio. The solid bars indicate that there was a layer present. The transparent ones indicate that there was no layer. And this effect seems to decrease as you increase the preload to comparable to what the uniform phantom was. So yes, as we increase amplitude, signal-noise ratio decreases. The signal-noise to ratio decreases, but that can be nullified by increasing preload. What is the impact of spatial frequency? So for 125 micron amplitude, there doesn't phantom with a variation of wavelengths. There doesn't seem to be any discernible trend across different spatial frequencies. So, and I also did this again for 500 micron amplitude. Again, there was no discernible trend between them. So it seems like spatial frequency doesn't make too much of a difference. You do notice the decrease in signal-noise ratio, but that's again with the increase in amplitude. And finally, we look at what the effect of using the compliant layer is. So comparing essentially the solid one with the transparent one behind it. And averaging across all scenarios, we do find that using the compliant layer increased the signal-noise ratio by about 22%. So that's sort of evening out of the stress field across the surface does actually increase the uniformity of the strain response. And there wasn't found to be any significant relationship for amplitude or spatial frequency for using a layer. Now we look at quantitative imaging. Again, this is just the uniform scan. So I can have a look comparison. This is the relative difference one. So again, comparing of elasticity at a specific point to all of the ones below it. So what these sort of darker ones indicate is that the measured elasticity here is higher than that as compared calculated directly from where the layer was. What we did notice though is based on the calibration curve, the mean elasticity within the region of interest does not follow what we expect. So up to about 30%, it's relatively accurate for elasticity, but beyond 40% in the hyperelastic region, you see a really large difference between the expected elasticity based on the calibration and that within the region of interest. And this was found to not always be the case. So just a rough comparison with the 125 micron amplitude phantoms, you see that for some of them, the elasticity, the measured modulus, did actually get up to around the 180 kilopascal range that we expected. So what's likely happened here is there's some experimental error, and I'll talk briefly about that later. It's likely due to friction between the stress layer and the imaging window. So we go back to answering our specific questions. So what is the impact of amplitude on the quantitative imaging? So you can see that at low preloads, the relative difference is significantly overestimated. So we find that within these sort of high amplitude phantoms, we're overestimating the elasticity within our region of interest. And that decreases as we increase preload. From up to about 60%, it's under 17%. This is because, so as we look at, this is our largest amplitude phantom. So remember these higher areas of strain that we had before. So because there's a higher area of strain there, it looks like there's a lot of force around that area. And so that it's like because there was a lot of movement, we estimate this as a softer region, whereas everything below it is overestimated. And these get included within our region of interest. So it's due to that sort of impact, and so which is why you see it decrease over time, over compression. So yes, at low preloads, we overestimate the stiffness of the material, and then that could be reduced by increasing the preload. Between amplitudes, it sort of evens out as you get to higher preloads, so there's no specific pattern between them at really high preloads. And now we look at the impact of spatial frequency. So going back to this image I showed you before of the 125 micron amplitude phantom, so you see really high measured stiffnesses, mean stiffnesses, which is a crude metric because there is an uneven stress distribution. But we wonder if this might have an impact on the relative differences within the phantom, and you find that it doesn't. So it does seem that although there is friction, the relative differences between the phantoms, it seems to be relatively negligible in this case. Again, as we increase amplitude, there's a very rough trend of decreasing relative difference, so it seems to be actually better in some cases, but increased wavelength, but it doesn't, it's sort of negligible. I think that I can't prove statistical significance with this because there's a relatively limited range of measurements, but we can provide rough trends. And this was similar for the larger amplitude phantom as well across a range of wavelengths. Again, that overestimation is just because of those friction, those high strain areas as we saw before. So again, there's no visible trend for variation in spatial frequency. It doesn't seem to change much, but increasing preloads does correlate with an improved accuracy of the measurement. So this is just summarizing the results. So in strain imaging and quantitative imaging, the impact of amplitude is more significant than that of spatial frequency. And using a compliant layer for strain imaging does improve signal-noise ratio by about 22%. So talking about my project itself, so what I've really done is I've set up a framework for future investigations of how to sort of analyze the quality of these images and the impact specifically of surface roughness. So one of the problems I mentioned before is friction. So by having a lot of friction between the stress layer and the phantom, it didn't really make much of a difference because the phantom and the material are made of the same, they're homogeneous. So it becomes just one solid mass. Preliminary results for ex vivo scans is that the stiffness of the stress layer is actually, it's more stiff than the breast tissue itself. And as you increase preloads, you increase the size of these gaps. So there does need to be an investigation there. So overall, the amplitude of the asperities seem to have a larger effect than that of spatial resolution, spatial frequency. And so further investigations must be carried out before any specific recommendations on how to image ex vivo or in vivo scans. So future work. So as I showed you before, that friction between this imaging layer and the imaging window and the stress layer, so all of those scans should be redone with improved oil lubrication between them, because it was found to be not the case in all of the scans, so there was clearly some experimental error there. As I mentioned before, we're really just looking at the coupling between the stress layer and the compliant layer for two equal materials in this instance. But it now makes it quite easy to do comparisons between either a stiffer or softer phantom, depending on what sort of investigation you're hoping to carry out. Another point would be to look at tumor mimicking phantoms, inclusion phantoms. So if you find, because tumors are heterogeneous and a bit more stiff, so putting it at different points within the phantom to see, could you detect a tumor under a point of high stress versus low stress? Does that make much of a difference? And that's where I see this work going. And yeah, thank you to my supervisors and the whole Bright Lab team and Pejman, for being there for whenever I last minute try and need some help. All right, thanks everyone.
Any questions from Zoom? Any questions from Zoom?
Have you turned yours on?
Any questions from Zoom? Sorry. Okay, probably is Rebecca is making noise. So that's okay. So any questions from here?
Thanks, Glenn. That was really nice.
Thanks.
So I have two questions for you.
Mm-hmm.
One is you relied on your edge detection method.
Yeah.
So how much are your results dependent on the accuracy of your edge detection?
Yeah. So that's a dependent question on how much compression we have. So if you look back at, it was this slide here. So what edge detection sort of is we use that as a measure of strain. So at really low compressions, the deviation in edge detection accuracy doesn't have nearly as much of an impact on because the Young's modulus is just, the elastic modulus is just a tangent here. But as we sort of increase compression beyond a point, edge detection becomes significant. So it's very dependent on how you actually take the images. For my situation where the layer thickness was about 500 micron, and in the total sort of imaging space, it's the imaging region in OCT is about 2.5 mil. So you can change it you can minimize that effect by having a larger phantom or a larger stress layer or lower preloads. So it becomes quite like a nuanced question depending on how you actually sort of take that image.
And then did you have to do any investigation on how the result of your work is affected by the accuracy of your
At the edge detection itself? I did do some investigation into that, and I found that it's within. So in this case, it was within about 10 to 20 micron. Didn't seem to have much of a. So you could see differences in the edge detection, but the final the impact was minimal. Yeah.
And the other thing is, so how long do you think it will take to get this in the clinical?
So there is a handheld probe being sort of designed. I think the researchers, they're actually doing the scans at the moment in surgery interoperatively at the moment. Yeah, so it's happening right now, I think. Yeah.
The other question I had you answered, it was
okay.
about the tissue differences in real patients compared to a phantom.
Okay. Yeah.
So, well,
Yeah.
Okay. Any other questions? Nothing? So we partly answer our question at the end about the depth of the tumor.
Yeah.
So which level of confidence you can detect the tumor?
So because this is just margin assessment, so the idea is to take the image after the bulk of the tumor has been removed, and you're just checking around the cavity to see is there still tumor there. OCT imaging systems, it's about up to 2.5 mil, where you can see depth, but as the signal to noise ratio for the OCT decreases, the accuracy of the strain image decreases. So it tends to be that in the current protocol, we only image up to about 500 to 800 micron, so half a mil to 0.8 mil. Yeah.
So detecting even tumor beds and
Yeah, something like that. Yeah.